Okay, welcome everyone again to the Green Ear Hub webinar series. My name is Barry Crosby and I'm one of the communication officers for the Green Ear Hub project. If you've been here with us before, you'll know that the Green Ear Hub project officially started in September 2022 as a coordination and support action of the European Commission under the Horizon Europe programme, and it currently represents 15 EU ERA-NETs, co-funds and self-sustained initiatives in the field of agri-food and biotechnology. We started these breakfast clubs here in October last year, and we've had 4 very successful series so far. The first was Circularity in Agriculture. The second series was related to biotechnology to advance sustainable solutions for bioeconomy. And the third was Sustainable Food Systems. And the one that we just finished at the end of 2024 was Precision Agriculture Technologies for a Sustainable Future. If you're interested in listening to the talks related to any of the topics from the series just mentioned, our recordings are now available to view on our YouTube channel. For the past 2.5 months, we've been focusing on the topic of plant health and protection, where we've hosted a series of expert-led discussions aimed at fostering a deeper understanding of plant pathology, integrated pest management, and sustainable agricultural practices. And just a few housekeeping announcements. Please do feel free to type your questions in the Q&A chat section, which I've enabled for everyone this morning. I'm going to do my best to address as many questions as possible within the time that we have today, which is 1 hour. If at the end of the session you have questions for either of our speakers, please do remember to address the speaker in the question. Now, today we have 2 panelists joining us, Dr. Giel Pell and Evelyn Everard. And we'll open our session this morning with Dr. Pell, a distinguished phytopathologist whose research focuses on plant health, particularly in the context of sustainable agricultural systems. A scientific officer at the Netherlands Food and Consumer Product Safety Authority, Dr. Pell leads and coordinates interdisciplinary research projects aimed at addressing critical questions relevant to policy making in the field of plant health. His collaborations extend to national and international universities, research institutes, and diagnostic laboratories, where he contributes expert insights to inform and guide policymakers in the development of effective agricultural strategies. Dr. Pell has authored numerous publications in peer-reviewed journals, elucidating the intricate mechanisms of plant immune responses and microbial interactions. Today, Dr. Pell's talk is entitled Taxonomy and Epidemiology of Pectobacterium and Dickeya species in Europe, North America, and South Africa, which is part of the UFresco Phytosanitary Research Initiative. Dr. Pell, thank you very much once again for accepting our invitation for our webinar series. I'm going to stop sharing my screen now so you can start sharing your presentation. Thank you very much.
Barry, for this kind introduction. I will share my screen. Thank you very much. Thank you very much for the invitation to give my talk here today. My name is Giel Pell. I work at the National Institute for Vectors, Invasive Plants and Plant Health. We call it the NIVIP. And we are part of the Netherlands Food and Consumer Product Safety Authority, the NVWA in Dutch, and also the National Plant Protection Organization of the Netherlands. So we are a governmental organization. And before I continue to anything else in my talk, I want to stress that Wageningen is not a synonym for Wageningen University and Research, because we are also based in Wageningen. Many times when I say, oh, you're in Wageningen, people think I'm from the Wageningen University. I get introduced into talks like this, and I think, no, that's not true. So Wageningen University and Wageningen is something different. And here you see in this picture, do you see my mouse? I don't think so. I'll get a laser pointer. Yes, now you see. So here is the Wageningen University in Wageningen, and we are here. So completely different, just so you know. So just before I start my talk, just a little bit about what we do and who we are at the NIVIP. We're mostly a diagnostics lab with around 110 people working there, divided in different groups based on the topics we work on. So we have bacteriology, where I work, but also the other groups, and very important groups, the molecular biology group, who supports all the other groups with molecular work. And we work on all kinds of topics. So yeah, we're mostly a diagnostics lab, like I said, but we also do research activities. And this is not the kind of research performed at the university. So no fundamental research, protein-protein interaction, stuff like this, no effector proteins. This we don't do, but we focused on, well, actually the data we need at our organization. So as a diagnostics lab, we need diagnostic protocols and tools, so we develop these. We also perform phylogenetic research and also epidemiologic research. And this is because we need to know the organisms we are working on. We're focused on detecting quarantine organisms within the European Union. So that's our main task. So we need to know a lot about these organisms, which kind of variants there are, but also what risks they pose to the farmers, etc., and how they spread, things like this. And that's also because we have an advisory task to the policymakers. So if there is an outbreak of a certain bacterium, they need to know what to do, and we have to advise them on this. So this is the kind of research we perform. And now my slides are not moving. Yeah. And one of these projects we performed was the project I'm going to present today. So this is also, our work was also part of this Refresco project. It was a 2-year project that started in 2021, so it finished in 2023. And it involved 15 organizations, which I listed here, but I'm not going to name them all. The work I'll present today is our work, which we brought into this project. We don't have time to address everyone's work today. And we focused on bacterial soft rot. Here you see a few pictures of bacterial soft rot on different hosts, cabbage or potato, bell pepper, tomato. And I think one thing you can see in all these pictures, you don't want to eat this. They don't look very nice. So this is, all these pictures were caused by bacterial soft rot. The bacteria causing these diseases cause more crop loss than any other bacterial disease known, and they can affect plants in a field or greenhouse or while growing, but also food that is stored. So even after harvest, these bacteria can affect these plants. They mostly affect succulent plant parts, so they need, let's say, juicy bits. And they affect a wide host range. Nearly every plant family is affected by these bacteria. Then they're also present around the world, everywhere, a wide range of temperatures. The optimum is 20 to 25 degrees, but at much higher or lower temperatures, they can also cause problems. One thing they do need is wet conditions. So if it's really dry, you will not have a lot of problems with these kind of bacteria. And then the symptoms, you already saw them in the pictures I showed. But you get these water-soaked spots like here in this picture. And if it goes further, then the discoloration continues and gets soft and mushy. And if you even wait longer, then moisture starts to drip from it. It will be really a messy thing. I think this is this kind of disease that is well, let's just say most disgusting you encounter with bacteria. Yeah, then this disease can be caused by many different bacteria, actually. I think the most important group are the Pectobacterium and Dickeya species, and that's also what I will focus on today. Yeah, so these two genera are part of the Pectobacteriaceae family. And here you see them with a box, and I also will talk about the Musicola. I will tell you why I included this as well in a minute. Here you see the list of Pectobacterium species. Actually, this was the list when I made the slide back in 2023. I checked last week, and there's 2 more new species. And actually, if you look at this list and look at the dates the species were published, there were a lot of species that were published relatively recently, so 2019 here. 2019, 20, 22, 19, 18. So a lot of new species. So in the recent years, a lot of has changed within this group. And also for Dickeya, you see actually the same thing. There are 12 species back in 23, and I checked again, but still 12 species. So no new ones here, but also quite a few very recent species. And then the Musicola, which I mentioned, this was one of the new species. Actually, when we started the project, it wasn't published yet, but at the beginning of the project, it was published. And this was a Dickeya paradisiaca, which they decided it was not a Dickeya, but it should be a Musicola, and they described the new species and included the paradisiaca in a new genus. So these are the species we work on in this project. And why do we work on this on these species? Well, I think the main reason is that they cause this disease, the backlag of potatoes. So this is a bacterial soft rot disease. It affects potatoes. You see the typical black leg here on this picture. So it affects the parts underground, so the potatoes, but also the first part of the stem, and the plants just fall over and die very quickly after you see the first symptoms. What is interesting about this disease, I think it's caused by many different species. So here you see the most important ones in Europe. So that's Dickeya solani and the Pectobacterium brasiliense. But in the US, you see different species is the most important one, but there are other species. And these species or this disease causes an annual loss of around 12 million euros in the Netherlands alone, and then only on seed potatoes. So not taking into account any of the other potato industry. Then this is data from the NAC in Emmeloord. They are the Dutch inspection service focusing on potatoes. And they were also part of this Refresco project. And they checked what kind of species they find on diseased potato plants. And they monitored this for years. If you look at the axis here, it's not linear, so pay attention to this. But what you see is that in the beginning of the 2000s, there were 3 different groups, actually, all of them around 30% present. And then in the later years, you see that the Dickeya species really increased in numbers. So most of the diseased plants, most of this disease was caused by the Dickeya. But then starting at the 2010, I think, and then continuing in the years that came after, you see a strong increase of the Pectobacterium brasiliense up to almost 100% in 2021. So you see that it's a really dynamic field. At first one species is important, then it changes to another one. And these are the most abundant species, but there are more species that can be found on the potatoes in the Netherlands. And here you see a list. Also from the NAC, you see Pectobacterium brasiliense is the most important one, but also a lot of isolates were belong to Pectobacterium species. So this means they didn't identify them further, but it's not one of these, but one of the other species. But you see many different species being present and causing problems. And this actually made us wonder what is going on. So these pathogens are not quarantine organisms, so they are not our main focus in our lab, but because they are quarantine organisms in some third countries, we get questions about them, and we need some information to advise the policymakers as well. And we actually didn't really know what was going on. We see a lot of species, but we don't know which species we have in the Netherlands. We don't know which species we import in the Netherlands. So we didn't really we were not able to assess the risks of these bacteria. So we decided to look into our collection. We had around 400 isolates coming from samples collected since the 80s, from plants grown in the Netherlands, but also from import material. But in most cases, they had old names like Erinea chrysanthemi or Erinea cultivora, which the species don't exist anymore. Or we identified them as Pectobacterium species, Dickeya species. So we decided to do something about this. And we selected 70 isolates that were sampled in the Netherlands, like I said. but could also be on import material. So not all of them are present in the Netherlands, let's say. We collected all the type strains of Pectobacterium and Dickeya, and we performed Illumina sequencing. So we sequenced these 70 isolates, and we also sequenced the type strains of which no whole genome data was available at NCBI. So we sequenced around 80 isolates. We performed de novo assembly, and then we performed the fast ANI, and we got this tree. I think there's a lot of data in one tree, so I split it in two halves. This is the bottom half. These are the Pectobacteria. In the boxes, you can see the type strains. And if the box is red, like here, it means we have isolates that are of the species. We found them in our collection. If they're green, we didn't find them. And you see, we actually find most of the species in our collection. So this means they are present in the Netherlands or could become present in the Netherlands because of import. One thing or two things I want to point out. You see this isolate, for example, that doesn't belong to any of the type strains because it's closely related to Pectobacterium parvum, but actually this type strain is more closely related to this type strain. So this is something different. And here we have a whole group which does not belong to any of the type strains. Besides identifying existing species, we also find some non-existing species, let's say. Then the Dickeya part get the same ID, so we find a lot of different species. No new species here, but we have a lot of variation in the Netherlands. And then, of course, you wonder, okay, you find a lot of different things. Is this a problem? And to test this, or to at least get some idea about this, we did some inoculation experiments with these isolates. So we inoculated potatoes by injecting them with a suspension of bacteria. And then we took part of these potatoes and planted them in pots and placed them in the greenhouse and scored the disease on the plants that came out of these potatoes, or in some cases, the potato never grew. And we also stored them in the stove at a warm temperature to see if you store them, if the bacteria cause problems. And then we get this as a result. So this is the same tree again, so with the Pectobacteriaceae and the Dickeyas. And then we selected isolates throughout the tree, and these are marked with the gray markings. And then you see the results of the stove experiment on the left side and the greenhouse experiment on the right side. So we categorized the symptoms. So from 0, here you see the legend from 0 to 3. So if it's yellow, it's healthy, and if it gets darker, the more dark, the more disease we found. And also these bars, they are the area under the disease progression curve. So it's kind of a quantification of the darkness of this picture here. So the higher the bar, the more disease we found. So above this dotted line here, you see the Dickeyas. You see most of them don't cause a lot of trouble, or they're not really, really highly virulent that they kill all plants. But you see that they mostly cause problems in the greenhouse experiments, while less problems in the stove experiments. For the Pectobacteria, you get the different view, actually. So you see a lot of symptoms in the stove experiments, less in the greenhouse experiments, except for this one. Let's show you in more detail. So here in the bottom, there are the controls. We included this Pectobacterium brasiliensis, of which we know it's very virulent in the field. So when we do the greenhouse experiments, on the right, you expect large problems with these bacteria. And actually, you see out of the 9 potatoes we put in the ground, only 2 came out, and they were dead after, let's say, 3 weeks. So they just came out of the soil and died. So it's very virulent. And actually here, this isolate is as virulent as this Pectobacterium. This Pectobacterium, you see only 2 plants coming out of the soil, and 1 died also very quickly, and 1 seems to escape everything. So maybe problem with the inoculation, but it's very virulent. And then if you look at what species this is one of the new species. So we don't know actually what this is. It came from a pumpkin kind of thing, or squash. So you see that this is an example of you find something we didn't know it existed. It comes from a non-potato crop, but causes a lot of can cause a lot of problems on potato. So you see that's important to look at this kind of things. So that brings me to the conclusions. We find a large diversity of Pectobacterium and Dickeya species in the Netherlands or imported material. Only a limited number seems to be highly virulent, so they're not all equally dangerous, let's say. And it's hard to link this virulence to phylogenetic position. So we were hoping to see like this whole group will be virulent and it will all be in one or two phylogenetic groups, but it's not like this. It's more spread throughout the tree. Yeah, that brings me to the acknowledgement slide. So I have to thank my team, so my colleagues, and especially Marisha, who was a student back in those days in our lab and performed all the inoculation experiments. And then, of course, the molecular biology team, who is heavily involved in all the sequencing we performed. And of course, the NAC who shared the data I showed today. And that brings me to the questions, I think. I will stop
Uh,
sharing my screen.
Aiel, thank you so much for such an interesting and insightful presentation. It's really given us an excellent snapshot, I think, of what you're trying to do with it and the results that it's yielding. It's very, very interesting. I just want to remind everyone that the chat box and Q&A section is open, so please do feel free if you have a question for Dr. Pell at any point to type that question in there. Dr. Pell, just while we're waiting for questions to come in, I was just kind of struck by your kind of discussion about the difficulty that you have in knowing what Dickeya species can be found, for example, in the Netherlands. And that leads me to the question of asking what, in your opinion, are the main challenges that you and your team would face in accurately identifying Pectobacterium and Dickeya species in plant samples? Like, is it just the lack of international collaboration and open science and information sharing? What are the kind of major obstacles, do you think, in terms of your sheer ability to be able to identify those particular strains?
I think for us, the first thing is priority because we focus on quarantine organisms. For these, we make PCRs and design everything that we are sure that we can, if it's there, we detect it. And we don't do this for these kind of bacteria. So that's a choice, actually, choice we make or my bosses make. So that's the first thing. And then what's, for example, with Pectobacteriaceae and also the Dickeya, there are quite a lot of species, and you see it changes a lot. So if you would want to design the PCRs for all these species, you need a lot of PCRs. You need to implement them and keep them, let's say, up and running the whole time. So that's kind of a problem. What we're doing now actually is we do more and more of these whole genome sequencing, and we're trying to set up a pipeline now that we just, if we find an isolate and we're not sure what it is, we can relatively cheaply perform the sequencing and we throw it in this pipeline. We don't need bioinformaticians anymore, but we just start the pipeline and we want that it identifies the species based on what is available in like at NCBI. So that's something. But I think that's the way to go for these kind of bacteria. But still, the price of whole genome sequencing, if you have a lot of them, can become very high. If you have a few of them in a year, it's not a problem. But if you have like 10 in a week, it becomes very expensive.
Yeah.
So then it's price. I think all the tools are there, all the techniques are there to properly identify them. It's just a choice, actually. Yeah.
Sure. Thank you very much. So just a couple of questions coming in from our audience. So very interesting with the large diversity found. Do you know anything about whether some of these work in synergy?
I do know we find them sometimes several species together, but then I don't know what this means actually. So I suspect in some cases they work in synergy, but that's just more of a hypothesis than something we tested.
Sure. And another question just coming in. Thanks for the great presentation, Giel. Are the genomes you sequenced published already?
The Pectobacterium we also use together with the Wageningen University in another project, and they're also already available at NCBI. The Dickeya, not yet, because we didn't publish it we didn't use it in a publication yet, but we are planning to deposit them at one point. Just not sure at which point.
Sure.
Right.
And another question, could you attribute the changes in species dominance over time partly to climate change impacts?
That's a good question. We talked about this also with the colleagues from the NAC, and we actually never came up with a good hypothesis we could test. So we're not sure, and then it's always very tempting to point at climate change because that's something that's changing. But I would say the changes in species is faster than climate change is going, so not sure about that.
Excellent. And another question, thank you for the interesting presentation. Are some species self-reliant, or do they interact with other species in the environment to be able to effectively cause the disease?
I think the highly virulent ones, they can cause problems all by themselves. The NAC also does a lot of work on the Pectobacterium brasiliensis species. And even within the species, you have highly virulent ones and non-virulent ones. And it seems to be related to the presence of a few effector proteins. They did some work together with the Wageningen University on this. And then you see that the specific weapons they have to attack. So at least for the highly virulent ones, I think it's just this species that can cause the problems. For the less virulent ones, maybe they need a nematode being present, causing some damage, and then they have an entrance and stuff like this. So.
Excellent. And one final one, Giel. Are there specific environmental factors that influence the spread of these pathogens in different regions?
Yeah, wet conditions are important. And actually, at the Wageningen University, Jan van der Wolf performed experiments on this, and they also tested the presence of these species on insects, for example. And they can find them on insects and also in rain, I think. So they are also present on the outside of plants, and then they are spread like this. I don't think they tested if they can also cause problems after being spread by insects, because of course the inoculation rates will be very low. There will be only a few bacteria spread, but at least they showed that they are spread like this.
Yeah, thank you so much for taking our questions. And I think thank you once again for such an excellent presentation. I think we could go on and on for another half an hour with questions on what really is fascinating research. Now, joining us
Yes.
is, thank you
Thank
so
you
much.
also. Yeah.
Thank you very much. I'm just going to share my screen now once again. And okay. So joining us now is Ellen Everhart, a prominent researcher at the Flanders Institute for Agriculture, Fisheries and Food, ILVO in Belgium, who specializes in phytopathology and plant disease management. Throughout her career, Ellen's work has focused on developing sustainable approaches to address key agricultural challenges that include the management of diseases affecting fruit and vegetable crops. Specifically, her research encompasses a range of initiatives that seek to strengthen collaboration in phytosanitary practices, emphasizing innovative methodologies for pathogen detection and control against plant diseases. She has authored several notable publications over the years that explore the biology and epidemiology of plant pathogens, reflecting her strong commitment to improving strategies for disease management in agriculture. Today, Ellen's talk is entitled Towards Improved Nepovirus Detection and Identification in Xiphinema Nematodes. And then thank you very much for accepting our invitation for a webinar series. We're absolutely delighted to have you with us today. So without further ado, the floor is yours. I'm going to stop sharing my screen now. You can start sharing yours.
So the research I will present today was performed in the framework of the Xiphinema project. It was a UFresco project together with many different partners. So as Barry already said, I work at Flanders Research Institute for Agriculture, Fisheries and Food. My main expertise is in plant virology under the supervision of Chris de Jonge. So let me start by a short introduction. So Xiphinema nematodes are migratory root ectoparasites. There are more than 250 species, of which approximately 50 belong to the Xiphinema americanum group. This group contains several quarantine species or Q species, such as Xiphinema americanum sensu strictum. Next to this Americanum group, there is also a non-Americanum group containing all other Xiphinema species. This group does not contain quarantine species. However, regulated non-quarantine species or RNQPs exist, and examples are Xiphinema index and Xiphinema diversicaudatum. So several Xiphinema nematodes have the ability to transmit Nepoviruses. The Nepovirus genus is a complex group of viruses. There are 46 species, which were formally divided or organized into 3 different subgroups, being subgroup A, B, and C. And examples of relevant species are, for instance, grapevine fanley virus, rabies mosaic virus, and tomato ring spot virus, all regulated non-quarantine pathogens. So while the direct damage caused by Xiphinema nematodes is usually limited, Nepoviruses can cause severe crop damage. So from left to right, you can see examples of crop damage caused by tomato ring spot virus, grapevine fanley virus, and rabies mosaic virus in tomato, grapevine, and hop, along with their important vectors. In trade, current procedures rely primarily on Xiphinema species identification. However, this process is challenging both morphologically and molecularly. And as a result, import is often held off solely due to the potential presence of certain species belonging to the Xiphinema americanum species group. So to overcome this, it has been suggested to only consider those Xiphinema specimen that are actually carrying quarantine viruses as a plant health risk. So certain currently quarantined Xiphinema species that are free of quarantine viruses could therefore be allowed to enter trade in certain circumstances. So as a result, focusing on direct detection and identification of Nepoviruses in Xiphinema nematodes could be an interesting alternative. So the main goal of the study was therefore to optimize and validate a generic method for Nepovirus detection and identification in Xiphinema nematodes. And to achieve this, we tried to optimize all steps in this procedure, including nematode extractions from soil, physical disruption, RNA extraction, and Nepovirus detection and identification in Xiphinema nematodes. And for the latter, we evaluated existing generic PCR assays, and we explored the potential of Oxford nanopore Menian sequencing technology, or in short, ONT. So to optimize nematode extractions from soil, we compared 2 different methods, the FLEC-modified COP method and automated zonal centrifugation, or the AZZ-C So while the FLEC-modified COP method is a manual decanting and sieving technique, the AZC is automated and uses zonal centrifugation with magnesium sulfate and kaolin as separation liquid and sediment film. So in this comparison, potting soil was used that was infested with Xiphinema diversicaudatum nematodes. For each method, nematodes were extracted from 20 randomly taken soil samples of 200 cc. Nematodes were counted visually, and then we distinguished intact specimen from damaged ones. And due to the design of the AZC, which only extracts half of the soil suspension, the results obtained with this method were doubled to compare them with the FLEC-modified COP method. So when we look at the results, we see that the AZC extracted almost double the amount of nematodes compared to the FLEC-modified COP method. And in terms of damaged nematodes, there were no damaged nematodes counted for the FLEC-modified COP method and only 6 for the AZC, which was considered as negligible. So based on this experiment, we would suggest that AZC is the preferred method. Then on this slide, we'll discuss the optimization process for physical disruption and RNA extraction. So nematodes, let me get my laser pointer up here. Yeah, it's difficult. Yeah, so nematodes consist of an outer tough cuticle, which contains collagen and which serves as a barrier during RNA isolation. This cuticle therefore needs to be disrupted. So for physical disruption, we compare 3 different methods. In the first method, nematodes were sliced in 3 to 10 segments, depending on the length of the nematode, and using a tip of a sterile needle. In the second and the third method, nematodes were bead-beated using a tissue lyser or a bead beater. And in the third method, the samples were treated with collagenase prior to bead beating to enzymatically break down the collagen. Then for RNA extraction, also 3 different methods were compared: a CTAP extraction with chloroform and isoamyl alcohol, a RNase plant minikit isolation using silica columns, and a RNA and a Kingfisher Magmax RNA isolation with a extraction robot and magnetic beads. So the different physical disruption and RNA extraction methods were evaluated over several experiments with different virus-carrying nematodes. But since the results were consistent across the different experiments and due to time constraints, I will only discuss those from the largest experiment with GFLV carrying Xiphinema index nematodes. So a total of 945 nematodes were divided over 3 replications, each consisting of 9 combinations of methods. So all possible combinations of physical disruption and RNA extraction. Then every combination of methods consisted of a negative control, a positive control, and the same combination of 18 samples as shown on this slide. And then the performance of each method was assessed using the qPCR according to CPAN, which was GFLV specific. So before discussing the results, it's important to note that the qPCR was validated beforehand and a CQ cutoff value was set at 37. Furthermore, all controls behaved as expected. So when we look at the 2 figures, the left figure shows the estimated mean CQ value, and the right figure shows the proportion of GFLV positive samples. both in function of the RNA extraction method and the nematode numbers and life stages present in the sample, where A stands for adult and J stands for juvenile. So first of all, there was no evidence for a model with physical disruption indicating a negligible effect of this factor. So slicing, bead beating, and bead beating with collagenase didn't affect the CQ value differently. Secondly, for RNA extraction, both the estimated mean CQ evaluation and the proportion analyses identified CTAP as the least performing RNA extraction methods. The Kingfisher and the RNeasy were both well-performing methods, with the Kingfisher scoring slightly better than the RNeasy, but by a non-significant difference. Then thirdly, for nematode life stage, better results were obtained for adults than for juveniles. And then finally, for the nematode number, for samples with 3 adults, this detection was possible with 95% certainty, regardless of the RNA extraction method. For samples with only 1 adult, this was only the case after an extraction with the Kingfisher. Then for juveniles, on the other hand, very poor results were obtained for samples with 1 or 3 juveniles. Nevertheless, when we increased this number to 10, the performance also increased and even approximated those obtained by samples with 3 adults. And then the final goal of the study was to optimize Nepovirus detection and identification in Xiphinema nematodes. So first of all, we wanted to evaluate the performance of existing generic PCR and qPCR assays. This assessment was done by comparing the sensitivity of different generic assays with each other and with species-specific assays using 2 different approaches. So first of all, by analyzing a 10-fold serial dilution of a positive control consisting of RMV-infected leaves. And then secondly, by analyzing Nepovirus-carrying Xiphinema nematodes. So in the first step, 6 assays were selected after a thorough literature screening, 2 of which were generic and 4 were RMV-specific assays. So when we look at the results, we see that the generic PCR assay developed by Wei and Clover failed to amplify any signal when analyzing the RMV dilution series. The analyses were repeated several times with different annealing temperatures, but without any success. In contrast, the subgroup A test by DGRO successfully detected RMV with a limit of detection, or LOD, of a 10-1 dilution. However, the 4 RMV-specific assays were more sensitive and showed an LOD of at least 10-2 and even up to 10-4 dilution. Then in the second step, the performance of the assays was evaluated by analyzing Nepovirus-carrying Xiphinema nematodes. The generic PCR assay by DGRO was evaluated against single species assays for RMV, GFLV, and TEO-RSV. And when we look at the results, we see that while each specific single species assay successfully detected the target virus in the nematode sample, the generic PCR assay by DGRO failed to do so. And then the final aim of the study was to evaluate the potential of Oxford nanopore MNION sequencing for the detection and identification of Nepoviruses and Xiphinema nematodes. I will not discuss the method in detail, but very briefly, RNA extracts were selected from Nepovirus carrying Xiphinema nematodes. The library was prepared and loaded onto a MNION flow cell. And then finally, the data were processed through several bioinformatics steps. So when we look at the results, we see that ONT sequencing failed to detect GFLV and TORSV in a single nematode. Although reads corresponding to RMV were identified in the sample with 6 Xiphinema diversicaudatum nematodes, the low read depth and coverage impact the results' reliability. However, it should be noted that the RNA extracts were exposed to multiple freezing and thawing steps before sequencing, which of course may have contributed to these inferior results. So this brings me to the conclusion. The question was whether we can offer an optimized and validated method for Nepovirus detection in Xiphinema nematodes. Well, the answer is not yet. While we successfully optimized all pre-processing steps leading up to Nepovirus identification, we are still encountering some challenges with the actual detection and identification process. So for the pre-processing steps, the proposed procedures involve nematode extractions from sole using the AZC, physical disruption using any method investigated in this study, and RNA extraction using either the Kingfisher, Marmax, or the RNeasyBlend minikit methods. And then for Nepovirus detection and identification, currently only species-specific PCR and qPCR assays were able to reliably detect Nepoviruses in Xiphinema nematodes. The question is then whether we should prioritize the further development of new species-specific PCR and qPCR assays. So first of all, existing generic PCR assays fail to detect Nepoviruses altogether. Nepoviruses are known for a high genetic diversity and complexity, making the development of generic, robust, and sensitive assays for all species and isolates extremely challenging. Therefore, we also see little potential in the development of new generic assays. Then for species-specific assays, although they seem to yield good results, we would not recommend using them in a screening for all possible Xiphinema transmitted Nepoviruses. In such cases, plant pathologists would be forced to run various species-specific assays in parallel. And furthermore, each test would need to be pre-optimized and validated. And this is, of course, time and money consuming. And then finally, despite ONT showing somewhat better results compared to the generic PCR assays, it did not lead to the strong conclusions we were hoping for. Nonetheless, we did saw its potential and the power of this technology. And given the fact that it's also a rapid evolving field, we strongly believe in its future for Nepovirus detection in Xiphinema nematodes. This was my final slide. I would like to thank you for listening, and also I thank everyone at ILVO for helping me with this research.
Ellen, thank you very much for such an interesting and insightful presentation. I just want to remind everyone that the chat box and Q&A section is open, so please do feel free to type a question for Ellen at any point. Ellen, I was just wondering, just while we're waiting for a couple of questions and comments to come in, I was just wondering, in terms of epidemiology and transmission, I was wondering what environmental conditions favor the survival and transmission of Nepoviruses in Xiphinema nematodes?
Well, that's a good question. We also tried, while performing transmission assays with these Xiphinema nematodes, to, of course, optimize everything for a successful transmission of Nepoviruses by Xiphinema nematodes. Of course, the good temperatures, optimal temperatures, optimal humidity help for good transmission. I remember in the grow chamber, optimal temperatures were around 21, 23 degrees of Celsius. But that is, of course, Xiphinema and Nepovirus dependent.
Yes, thank you so much. Just a couple of questions coming in, Evelyn. Thank you for your interesting presentation. Could the detection of the Nepovirus be affected by the fact that the nematode might be non-living?
Well, the nematode was living when we extracted them. And then we, from the moment we extracted the nematodes from soil, we've isolated them in Eppendorf tube, and then we immediately tested them after storing them in a freezer for maybe 1 week or a bit longer. But when we extracted the nematodes, we made sure that they were alive.
Yeah, and I suppose just a kind of a follow on from that question, and I was wondering about how long can Xiphinema nematodes retain Nepoviruses, and does it differ between the various species, or?
Well, I am not sure, to be completely honest.
Yeah.
I do know that when juveniles go into mold, they lose the Nepoviruses because they are retained in the cuticle lining of the odontophore and oesophagus. So over the different life stages, they lose the viruses. But of course, when they feed again on the infected plant, they can acquire them again. The viruses also do not propagate in the nematodes. This will also play a role, but the exact time of how long they stay virulent, I don't know.
Yeah, and I was wondering, is there any evidence of specific interactions between Xiphinema nematodes and certain Nepoviruses that enhance transmission efficiency, I suppose, or more effectively?
So your question is whether for some Nepovirus Xiphinema combinations, the transmission was more successful than for others?
Yes, exactly.
I didn't observe that, at least with the species I tested. But in order to answer that question, yeah, I should do a more profound research or literature screening, or but to my knowledge, I don't think so, or I don't know at least.
Sure, of course, no problem. And then just one or two other questions. So somebody is writing, the EURL has recommended sorry, I don't know, my pronunciation of this is probably not correct. So TORSV specific real-time RT-PCR assay will to try to apply it for detection of TORSV in nematodes.
Yeah, well, back then that qPCR wasn't existing. It's dates back from 2020, I believe, the project. But indeed, we are now also using a qPCR for TORSV detection, but then in plant material because this project finished. But indeed, that's a very good alternative for the PCR assay I used.
Excellent. And just one final question. Why did you choose the MinION over Illumina sequence? And the depth of sequencing seems to be critical here.
Well, of course, in this project, or the tool that we wanted to use will be used in a diagnostic setting where you need your results rather sooner than later. So for this reason, the main advantage of MinION over Illumina is time efficiency. With MinION, you can basically have your results in real time, you have real time access to your results. Whereas for Illumina, we are working with an external provider, and there we need to wait at least 4 to 6 weeks before we have the results. So that's basically the main reason why we chose for Menayan.
Excellent. And the last question, I promise, again, just from the audience, can more than one Nepovirus be present in the same nematode? And can these be detected simultaneously?
Good question.
Yeah.
But again, I will have to disappoint the person who asked the question. I should check. I haven't seen multiple infections in Xiphinema nematodes yet, but if we want to know that for sure, we should test all 46 Nepoviruses with all 250 Xiphinema nematodes to know which combinations can exist and which cannot. But I don't know the exact answer to that question.
Ellen, thank you very much. I think we'll leave it at that point. It's just coming up on 10 o'clock. And thank you very much for your questions from the audience. And thanks so much to Ellen just for addressing those. So I'd just like to thank once again Dr. Hiel Pell and Evelyn Everard for giving us such wonderful and insightful presentations and providing us with tremendous overviews, I think, of their respective projects this morning. Our next webinar will be on April the 10th. We will have Katharina Einrich, an analytical chemist at Vera in the UK, discussing the use of stable isotope ratio analysis for the identification of invasive species native in alien environments. And Dr. Negin Ibrahimi, a senior researcher at the Flanders Institute for Agriculture and Fisheries and Food, ILVO, talking about the development of fast detection methods for quarantine Tifrite. Again, thank you very much to our panelists, and thank you everyone for attending this morning and for the excellent questions. We really hope you enjoyed the webinar, and we hope to see you back here on the 10th of April with us next time. Thank you very much.